In the Amazon rainforest of southeastern Peru, Gideon Erkenswick and his team are collecting biological samples from wildlife populations of bats and primates.
Regularly collect samples, blood tissue, buccal swabs, nasal swabs, and then release animals.
Erkenswick is part of the Division of Infectious Diseases at Washington University School of Medicine in St. Louis. He's also co-founder and director of Field Projects International, a research and conservation nonprofit. Portable sequencing technology is used to screen for disease-causing viruses associated with the movement of wildlife or wildlife products.
So COVID-19 is caused by a beta coronavirus termed SARS-CoV-2. Close to the area where we suspect the outbreak took place, there's a wildlife market.
Erkenswick is a postdoctoral research associate in the lab of Dr. Jennifer Phillips, co-director of the Division of Infectious Diseases at Washington University.
SARS-CoV-2 and related viruses circulate in bats, and how it actually spilled into people, still an area of active investigation.
Phillips is co-author of a study proving the value of broadly deploying new methods, including this next generation of sequencing technology. The team is extracting viral genomes from animal samples and rapidly sequencing them on site. The researchers are hoping to create a global wildlife surveillance system to identify animal viruses that can potentially cause human diseases. They say this new system could provide early warning for the next pandemic.
Kind of harness the capacity around the world that's not being harnessed with new technologies so that we can protect the whole world.
There isn't consistent pathogen surveillance taking place in wildlife markets or in wildlife communities or in peri-domestic animals.
The goal is to broaden wildlife surveillance globally, monitor hotspots and wildlife markets, and provide analysis and data collection with higher accuracy and greater speed than the way it has been done. They say they want to go beyond a process that's been unreliable with fewer available labs and resources.
This kind of system could produce data in a matter of days or weeks, whereas, prior systems that involve moving samples around, exporting samples, storing samples, finding the right labs to test the samples, that's years, months at best, sometimes years to achieve that work.
With this new way, viral sequences can be uploaded from on-site to a central database in the cloud.
It's not about building more high-tech labs distributed around the world. Cutting-edge technology that's more affordable and more portable that you can teach people on site to be able to use.
Collect samples and on the fly test for parasites or pathogens.
Researchers around the world could then help analyze them to identify animal viruses that may be a threat to people. Identification of pathogens would be more immediate.
We have to understand the human-wildlife interface better, so we have to make a science out of it that we don't have right now, and a science of trying to predict and understanding markers of where that risk really is.
Providing early warning for the next outbreak or pandemic so the world may be prepared.
What's out there so that should they spill, we could rapidly develop therapeutics that would encompass these other things that are in nature.
There's all sorts of natural sort of barriers when we sort of disturb those things by destroying habitat or removing animals from the wild. That's when exposure and transmission, which we never knew to occur, can take place. The world is very connected. People travel so easily now that there is virtually no place where pathogen, zoonotic viral surveillance is not a concern for us all.